Hello everyone, I am Aishi Datta from Ixpoe. Today, the topic which we are going to study is photosynthesis part 3. Energy efficiency of photosynthesis. The energy efficiency of photosynthesis is a ratio of the energy stored to the energy of light absorbed. The chemical energy stored is the difference between that contained in gaseous oxygen and organic compound products and the energy of water, carbon dioxide, and other reactants. The amount of energy stored can only be estimated because many products are formed and these vary with the plant species and environmental conditions. If the equation for glucose information given earlier is used to approximate the actual storage process, the production of 1 mole, that is 6.02 multiplied by 10 to the power 23 molecules, abbreviated as N, of oxygen and 1/6 mole of glucose result in the storage of about 117 calories of chemical energy. This amount must then be compared with the energy of light absorbed to produce 1 mole of oxygen in order to calculate the efficiency of photosynthesis. Light can be described as a wave of particles known as photons. These are the unit of energy or light quanta. The quantity n photons is called an Einstein. The energy of light varies inversely with the length of photon waves. That is, the shorter the wavelength, the greater the energy content. The energy of a photon is given by the equation E equal to hc by lambda, where c is the velocity of light, h is the Planck's constant, and lambda is the high light wavelength. The energy of E of an Einstein is E equals to Ne by equals to nhc by lambda, that is equals to 28,600 by lambda when E in calories and lambda is given in nanometers. Nanometer 1 nanometer is equal to 10 to the power minus 9 meter. An Einstein of red light with the wavelength of 680 nanometer has an energy of about 42 calories. Blue light has a shorter wavelength and therefore more energy than red light, regardless of the whether the light is blue or red. However, the same number of Einsteins are required for photosynthesis per mole of oxygen formed. The part of the solar spectrum used by plants has an estimated mean wavelength of 570 nanometer. Therefore, the energy of light used during photosynthesis is approximately 28,600 by 570 or 50 cal per Einstein. In order to compute the amount of light energy involved in the photosynthesis, one other value is needed. The number of Einstein absorbed per mole of oxygen evolved. This is called quantum requirement. The maximum quantum requirement for photosynthesis under optimal condition is about 9. Thus, the energy used is 9 into 50 or 450 cal per mole of oxygen evolved. Therefore, the estimated maximum energy efficiency of photosynthesis is the energy stored per mole of oxygen evolved 117 cal divided by 450, that is 117 by 450 or 26%. The actual percentage of solar energy stored by a plant is much less than the maximum energy efficiency of photosynthesis. An agricultural crop is which the biomass total dry weight stores as much as 1% of total solar energy received on an annual area weight basis is exceptional. Although in a few cases of higher yields, perhaps as much as 3.5% in sugarcane have been reported, there are several reasons for this difference between the predicted maximum efficiency of photosynthesis and the actual energy stored in biomass. First, more than half of the incident sunlight is composed of wavelength too long to be absorbed, and some of the remainder is reflected or lost in the leaves. Consequently, plant can best absorb only about 34% of the incident light, sunlight. Second, plant must carry out a variety of physiological processes in such non-photosynthetic tissues as roots and stems. This process, as well as cellular respiration in all parts of the plant, use up stored energy. Third, rates of the photosynthesis is bright sunlight sometimes exceed in need of the plant, resulting in the formation of excess sugar and starch. When this happens, the regulatory mechanism of the plant slow down the process of photosynthesis, allowing more absorbed sunlight to go unused. Fourth, in the many plant energy is wasted by the process of photorespiration. Finally, the growing season may last only for a few months of the year. Sunlight received during the other seasons is not used. Furthermore, it should not it should be noted that if only agricultural product that is seed, fruit and tubers rather than total biomass are considered as the end product of the energy conservation process of photosynthesis, the efficiency falls even further. Chloroplast, the photosynthetic units of green plants. The process of plant photosynthesis takes place entirely within the chloroplast. Detailed studies of the role of these organelles date from the work of British biochemist Robert Hill. About 1940, Robert Hill discovered that green particles obtained from broken cells could produce oxygen from water in the presence of light and chemical compound, such as ferric oxalate, able to observe as an electron acceptor. This process is known as the Hill reaction. During 1950s, Daniel Aron and other American biochemists prepared plant cell fragments in which not only the Hill reaction but also the synthesis of the energy storage compound ATP occurred. In addition, the coenzyme NADP was used as the final acceptor of electrons, replacing the non-physiological electron acceptors used by Hills. His procedure were refined further so that small individual pieces of isolated chloroplast membrane or lamella could perform the Hill reaction. These small pieces of lamella were then fragmented into pieces of small that they perform only the light reaction of the photosynthetic process. It is now possible also to isolate the entire chloroplast so that it can carry out the complete process of photosynthesis from light absorption, oxygen formation, and the reduction of carbon dioxide to the formation of glucose and other products. Structural features. The intricate structural organization of photosynthetic apparatus is essential for the efficient performance of the complex process of photosynthesis. The chloroplast is enclosed in a double outer membrane and its size approximates a spheroid about 2500 nanometer thick and 5000 nanometer long. Some singles called algae have one chloroplast that occupies more than half of the cell volume. Leaf cells of higher plants contain many chloroplasts, such approximately the size of one is some algal cells. Within thin section of chloroplasts are examined under the electron microscope. Several features are apparent. Chief among these are the intricate internal membrane, that is the lamella, and the stroma. The colorless matrix in which the lamella are embedded, also visible, are starch granules which appear as a dense body. The stroma is basically a solution of enzyme and small molecules. The dark reaction occurred in the stroma. The soluble enzyme which catalyzes the conversion of carbon dioxide and minerals to carbohydrates and other organic compounds. The capacity of carbon fixation and reduction is lost if the outer membrane of the chloroplast is broken, allowing the stroma enzyme to leak out. A single lamella, which contains all the photosynthetic pigments, is approximately 10 to 50 nanometer thick. The lamella exists in more or less flat sheets, a few of which extend through much of the length of chloroplast. Examination of cross sections of lamella under the section electron microscopes show that the edges are joined to form closed hollow discs that are called thylakoids. The chloroplast of most higher plants have regions called grana in which the thylakoids are very tightly stacked. When viewed by electron microscope at any oblique angle, the grana appears as stack of discs. When viewed in cross section, it is apparent that the thylakoids extend from one grana through the stroma into other grana. The thin aqueous spaces inside the thylakoids are believed to be connected with each other via the stroma thylakoid. These thylakoid spaces are isolated from the stroma species by the relatively impermeable lamella. The light reaction occurs exclusively in the thylakoids. The complex structural organization of lamella is required for proper thylakoid function. Intact thylakoids are necessary in the formation of ATP. Thylakoids that have been broken down to smaller units can no longer form ATP, even when the conversion of light into chemical energy occurs during electron transport in these units. Such lamellar fragments can carry out the Hill reaction with the transfer of electrons from water to NADP+. Thank you so much. If you like the video, please like, share, and subscribe to our channel. We have previously uploaded photosynthesis part 1 and 2. You are requested to please go and watch the other videos so that you can understand this video better. Thank you so much.